Have you ever tried to find like a tiny little detail in a super complex image?
Oh, yeah.
It's like searching for a needle in a haystack, right?
Yeah.
Now imagine that tiny detail could be the key to diagnosing something as serious as colorectal cancer.
Right.
We're talking about those microscopic images, the histopathology slides. They're packed with information about cells and tissues, and specialists need to be able to zoom in and out, analyze it all.
It's like a multi-layered puzzle.
Yeah, exactly. And that's what we're diving deep into today, this new computational method called SwinCup. We've got a research paper here that really breaks down how this technology works, and specifically how it's tackling the challenge of analyzing those histopathology images for colorectal cancer.
Sounds fascinating.
Yeah, it is. So our goal is to understand how SwinCup is using AI to make sense of these colorectal cancer tissue samples, and why that even matters in the first place. Let's start with the basics. Why is examining these microscopic slides so crucial for diagnosing colorectal cancer?
Well, right now, the gold standard for diagnosing colorectal cancer really relies on pathologists examining those tissue slides under a microscope.
Oh, wow.
Yeah, they're looking for those telltale signs, but it's a very intricate and subtle process.
I bet.
Tissues can have a lot of natural variation in their appearance and the patterns they form. And as you mentioned earlier, scale is a huge factor.
Right, yeah.
They might need to zoom in to see the fine details of a single cell, and then zoom out to see how those cells are arranged within a larger tissue structure.
It's not like looking at a regular photograph.
Exactly.
Where everything's kind of at the same level of detail. And I know these slides are being digitized now, which makes sense for computer analysis.
That's right.
But imagine that presents its own set of challenges.
It does. So this move to digital slides has opened up a lot of possibilities for using computers to help analyze these images.
Okay.
But the problem is that these whole slide images, or WSIs as they're called, are massive.
I can imagine.
Like 1,000 times larger than your typical digital photograph.
1,000 times?
Yeah. So you can't just feed that entire image into a computer all at once and expect it to process it efficiently.
Right.
The usual approach is what's called a patch-based approach, where you essentially break the WSI down into smaller pieces, like tiles, and then the computer analyzes each of those individual patches.
But then you lose the bigger picture.
Exactly. You miss that context, the relationships between different areas of the tissue. It's like trying to understand a painting by just looking at individual brushstrokes.
Yeah, you lose the whole composition, the artist's intent.
Exactly. And our source points out that even though there's been a lot of progress in using machine learning to diagnose diseases from images, this area of analyzing colorectal cancer, WSIs, hasn't received as much attention as other cancers like breast or prostate cancer.
Oh, interesting.
So there's a real need for more specialized approaches. And that brings us to this idea of transformer models, which our source highlights as a recent breakthrough in computer vision. Have you heard much about transformer models?
I've heard the term, but I'm not entirely sure how they work.
So transformer models have become incredibly popular in computer vision in recent years. They've shown remarkable success in doing things like identifying objects in images, segmenting different regions of an image, and even understanding the relationships between objects.
So they're really good at understanding the context of an image.
Exactly. And that's because of this mechanism called self-attention.
Okay, break that down for me. What is self-attention?
So imagine the model is looking at an image.
Okay.
Self-attention allows the model to figure out which parts of the image are most relevant to other parts when it's trying to make a decision or prediction about the image as a whole.
Oh, so it's like it's focusing its attention on the most important parts.
Exactly. It's learning these relationships, even between parts of the image that are far apart. Now, within the family of transformer models, there's a particular type called the Swin Transformer, which is what SwinCup is based on. And the paper really emphasizes its hierarchical structure and its use of shifted windows.
Shifted windows.
Yeah, it's a bit of a mouthful, but it's a very clever idea.
It sounds very visual, too, like you're shifting your view to get a better look at something.
That's a great way to think about it.
Okay, cool.
So the Swin Transformer is really good at capturing both the local details, those fine-grained features, and the broader global context of an image.
Okay.
It does this through its hierarchical design, processing the image at different resolutions. Kind of like how CNNs, those convolutional neural networks, work, but with this added shifted window attention mechanism.
Right. So it's building up an understanding from the small details to the bigger picture.
Exactly. Now, SwinCup takes the Swin Transformer and applies it specifically to the task of segmenting those histopathology images for colorectal cancer.
Okay, so it's tailored to this particular problem.
Right. Now, SwinCup uses an encoder-decoder architecture, which is a pretty common approach in image segmentation. You can think of the encoder as the part that takes the input image, extracts all those important features, and the decoder then takes those features and uses them to create the final output, which in this case is a segmentation map.
A map that highlights the cancerous areas.
Right. Yeah, it's essentially outlining the different structures within the tissue sample.
Got it.
Now, one of the key innovations in SwinCup is its cascaded upsampling decoder.
Yeah, the paper mentions that. What makes that so special?
Well, this cascaded upsampling decoder is really important for how the model combines all the features that the encoder extracts.
Okay.
It starts with those low-resolution feature maps from the encoder, which have more global information, and then it gradually increases their resolution, kind of step by step,
Okay.
combining them with higher resolution features from earlier stages of the encoding. This helps refine the boundaries of the segmented structures, making sure those fine-grained details aren't lost in the final output.
That makes sense. So it's like putting the puzzle back together, but with information from different stages of the analysis.
Exactly. And the results from testing SwinCup on those two datasets, GLA and CRAG, were really impressive.
Yeah, that's what really stood out to me.
The model significantly outperformed previous state-of-the-art methods.
The improvement was pretty dramatic, too, like around 23% in some cases.
That's right, which could have a real impact on how quickly and accurately colorectal cancer is diagnosed.
Absolutely. Okay, let's dive a bit deeper into the specifics of how SwinCup works.
Sure.
We've talked about the Swin Transformer, but what exactly is happening in that encoder stage?
So the Swin Transformer encoder starts by dividing that input image, that huge WSI, into these small, non-overlapping patches.
Yeah.
Like tiny tiles.
Okay.
And then each of those patches is turned into a numerical representation that the transformer can process.
Okay.
The real magic happens in those transformer layers where the self-attention mechanism is applied. But it's not applied to the entire image at once. It's applied within these localized windows.
So it's looking for relationships within these small local areas first.
Exactly.
And then expanding outward, kind of like that hierarchical approach we discussed earlier.
You got it. So this window multi-head self-attention, or WMSA, calculates how much each patch within a window should pay attention to every other patch in that same window.
So it's figuring out the connections between those little pieces.
Right. But to get that global understanding, they use this clever trick called shifted window multi-head self-attention, or SWMSA.
Ah, yes, the shifted windows.
In the next layer, the boundaries of those windows are shifted, so you get these new windows that overlap with the original ones.
Okay.
And this allows the model to learn relationships between features that were initially in completely separate windows.
So it's like it's expanding its field of view.
Exactly. And this is done in a hierarchical way, too, with what they call patch merging, where similar patches are grouped together to create larger blocks, reducing the number of patches and increasing the complexity of the information in each one.
Okay, so it's moving from those small, local details to those larger scale patterns. Now, what about that decoder? How does it take those encoded features and actually produce the segmentation map?
So the cascaded upsampling decoder, or CUP, essentially reverses the process of the encoder. It takes the multi-level feature maps from the Swin Transformer encoder, starting with the lowest resolution maps that have the most high-level information, and then it progressively increases the resolution through a series of upsampling steps.
So it's going from that abstract representation back to something more concrete.
Exactly. And to make sure those fine-grained details aren't lost, it also uses skip connections,
Okay.
which take feature maps from earlier layers of the encoder that have more spatial detail but less high-level understanding, and combines them with the upsampled features.
Okay.
It's a way of bringing together the best of both worlds, the big picture and the fine details.
And that results in a more accurate segmentation map.
Exactly. Now, another really important aspect of SwinCup, and actually many deep learning models in medical imaging, is this idea of pre-training.
The paper mentions that.
Yeah.
So what exactly is pre-training and why is it so crucial here?
Pre-training is essentially where you train a model on a large data set for a more general task before you fine-tune it for your specific task.
Okay.
Think of it like learning the alphabet before you start writing novels.
Okay.
And the reason it's so important in medical imaging is because we often don't have a lot of labeled data.
Right. getting those expert annotations for medical images is expensive and time-consuming.
Exactly. So pre-training allows the model to learn general visual features from a much larger data set, even if it's not from the same domain.
Okay.
And these pre-trained weights can then be used as a starting point for training on the smaller medical data set.
So it's like giving the model a head start.
Exactly. And the paper explores both supervised and self-supervised pre-training.
Right. I was curious about that. What's the difference between those two approaches?
So in supervised pre-training, you're using a data set where each image has a corresponding label, and the model learns to associate those images with their labels. But in self-supervised pre-training, the model learns from unlabeled data.
Okay.
It learns by trying to solve some kind of puzzle that's inherent in the data itself, like predicting a missing part of an image or determining if two distorted versions of the same image are actually related.
So it's learning without explicit labels. That's pretty amazing.
Yeah, it is. And the paper specifically highlights this method called Momentum Contrast V2, or MOCU V2, for self-supervised learning.
Okay.
The idea is to train the model to recognize different views of the same image, even if they have slight variations.
So it's learning to be robust to those variations.
Exactly. And they actually modified MOCO V2 a little bit to account for the fact that in histopathology images, different patches within the same image might belong to the same class.
Right, right.
Which isn't always the case in the natural images that MOCO V2 is usually used on.
So a clever adaptation for this specific type of data.
Exactly. So what did they find when they tested all of this?
That's the exciting part.
As we mentioned, SwinCup consistently beat those existing state-of-the-art models on both the GLAS and the COG datasets. It achieved those high F1 scores, but also performed better in terms of recall and precision.
Right, right.
Now, when they looked at the impact of pre-training, they found that supervised pre-training on a relevant data set, in this case, one with prostate cancer histology images, gave the biggest performance boost.
Okay.
Pre-training on a larger but very different data set, like one for understanding everyday scenes, also helped, but not as much.
So the relevance of the pre-training data is crucial.
Absolutely. And their modified self-supervised learning approach also showed a significant improvement, which is really exciting because it means you don't necessarily need tons of labeled data.
Right. That's a big advantage.
Now, they also did an ablation study where they removed or changed different parts of the model to see how it affected performance. And this really highlighted the effectiveness of that cascaded upsampling decoder.
Yep.
And finally, they looked at the impact of the pre-training data itself. And they found that while large data sets can be helpful, using too much data from a completely different domain might not be optimal.
Okay.
And having more diverse medical images for pre-training generally led to better results.
So it's all about finding that balance between the size and the relevance of the data.
Yeah, exactly.
So we talked a lot about the technical details of SwinCup.
Right.
But why should someone who isn't a computer scientist or a pathologist care about this? What are the broader implications of this research?
Well, the real significance of SwinCup is its potential to improve the diagnosis of colorectal cancer.
Okay.
It's better at understanding those complex patterns in those histopathology images thanks to that global self-attention mechanism.
Right.
And because it can analyze those images more accurately and efficiently, it could help doctors make faster and more informed decisions about treatment.
So it could potentially save lives.
Exactly. And what's also really exciting is that the same principles behind SwinCup could be applied to other types of medical data, like CT scans or MRI images.
So this could be a blueprint for developing similar AI-powered diagnostic tools for other diseases as well.
That's right.
So to sum up, what would you say is the most important takeaway from our deep dive into SwinCup today?
I'd say it's this. SwinCup represents a really significant step forward in using artificial intelligence to analyze colorectal cancer histopathology images.
Okay.
And it does this by combining a clever architecture, the Swin Transformer and cascaded upsampling decoder, with really smart pre-training strategies. And what that means is faster, more accurate diagnoses, which could lead to better outcomes for patients.
Absolutely. And on that note, here's a final thought for you to ponder. SwinCup has shown us what's possible when we combine advanced AI techniques with a deep understanding of the problem we're trying to solve.
Right.
So what other areas of medical image analysis could benefit from similar approaches? Think about all the diseases that rely on interpreting medical images for diagnosis, from cancer to heart disease to neurological disorders.
It makes you wonder.
Could advancements like SwinCup pave the way for more accurate and efficient diagnoses across a whole range of medical challenges? The potential is definitely there.
Yeah, it's really exciting to think about
It is.
where this could all lead.
It really is.